In this video, we're going to cover the limbic system, which for the most part deals with memory and emotion, but because of sort of the origins of the nervous system, it also deals with olfaction. And this makes some good sense. So memories and emotions are there to guide our behavior. Do we go toward a certain area or do we avoid it? Well, if it gives us a reward, if it makes us happy, if we can find food there, if we have a memory of finding food there, we're more likely to go toward that direction. If there's something dangerous, if we've hurt ourselves, if it's uncomfortable, if somebody's a jerk over there, then we remember it didn't feel very good to go there, and so we move away. Now, the earliest animals were largely interacting with the world using chemical sensations. So that's why the olfactory system is going to have this ancient and direct link into the limbic system. Now, I will tell you, as primates, we also have a ton of visual input into our limbic system. So don't think that only olfactory information goes there. It's just that quite a bit of it does. And when it comes to storing information, that's going to happen here in the hippocampus. That's what we're looking at here. And this is from the rainbow mouse. And you can see why it's called that. In the brain, you see a rainbow of colors because of some random expression of fluorescent proteins. And we can see this is a structure that's jam-packed with neurons. It doesn't have your typical 6 layers. In fact, this is a 3-layered older version of cortex. So we're not in the cortex yet, but we're getting there, and we're most certainly going to talk to the cortex. So we'll start off with olfaction and how that thing works, move into memory, and then hit emotion in the end. And keep in mind, all of these things are here to guide our behavior. And so we're going to talk to things like the basal ganglia, right? And we're going to talk to things like the cortex. And that's the whole point. Make sure we're doing the right thing, something that has been beneficial in the past, and we avoid things that weren't beneficial in the past. Okay, so olfactory systems here, we're going to project pretty directly to the cortex. In fact, we're not going to go through the thalamus first. We're going to go to the cortex, then the thalamus, and then back to the cortex. So a little different here. All the other senses go through the thalamus first, but not the olfactory system. So our journey begins in the nasal cavity. There we've got some respiratory epithelia, which we don't care about. We do care about the olfactory epithelium, and that's right up in here. And we call it that because that's where the olfactory nerves live. So those olfactory sensory neurons, those are the primary afferents, and they make the shortest cranial nerve. There it is right there, just passing through that little bit of skull, that cribriform plate. That's cranial nerve 1. And we're going to form synapses here in the olfactory bulbs. So the olfactory bulb is not cranial nerve 1. No, these are the secondary afferents. Okay, so olfactory epithelium has the olfactory sensory neurons, and the olfactory nerve connects those neurons to the olfactory bulbs. So those olfactory sensory neurons, now we've kind of flipped so that up is down. So they're dangling their dendrites here. This is the olfactory cavity here. So here's where the air comes in, and there's these little molecules, odorants, that are diffusing in the air, and they hit this mucus and they diffuse in there, and they encounter the dendrites of olfactory sensory neurons. Each of these olfactory sensory neurons expresses what we call an odorant receptor. These are metabotropic receptors that bind to one or several odorants. When the odorant binds, it turns on the olfactory sensory neuron, and that's going to communicate via cranial nerve 1 to the olfactory bulbs. Now, these olfactory sensory neurons are exposed to air, as you can see. Yeah, they're usually covered with mucus, but there's some air up there, and so these die off. So every 1 to 12 months, we have to replace each of our olfactory sensory neurons, and that's why we have a whole bunch of what we call basal cells. And so as we lose our olfactory sensory neurons, these stem cells divide asymmetrically and make new olfactory sensory neurons. So we're always replacing that first cranial nerve. What happens when an odorant bumps into an odorant receptor is, of course, some G protein signaling. Now, I know they write golf here. That's just branding, ladies and gentlemen. That's GS, no matter how you cut it. Because what does it do? It turns on adenylyl cyclase. And so we make cyclic AMP. Cyclic AMP opens up those cyclic nucleotide gated cation channels that we've heard about time and time again. Yes, there's some other things happening. We got some calcium gated chloride channels that let chloride leaves, and that depolarizes us too. But that's enough. We open up a cyclic nucleotide gated cation channels, and by golly, we depolarize. And that is the basis of olfactory sensory neuron activation. Once activated, these olfactory sensory neurons, you'll notice they've now color-coded. They project into the olfactory bulbs, and their axons converge in these things called glomeruli. Here they are in cartoon format. And notice, the different colors here are all converging. Now, the color is showing us which odorant receptor is expressed. So if you express the same odorant receptor as your neighbor, you're going to project to the same glomerulus. And here it is in real life. Here they've done some beta-gal staining, and you can see all these axons converging, right there. To give some perspective, here's our olfactory bulbs down here. We got a couple glomeruli formed in each. And this is where all of the olfactory sensory neurons with the same odorant receptor converge. So this is my smell map. We can't make sense of it at this point, but clearly there's an organization, and that would be the glomeruli. So what we're bumping into there would be the dendrites of the secondary afferent, the mitral cells. The glomeruli is where we're going to pass the baton from primary olfactory sensory neuron to secondary mitral cell. Now, between the glomeruli, we're also communicating. We have to clean up the signal because the projection is pretty good here, but it ain't perfect. And so we have lateral inhibition. And it takes place at two levels. There's the periglomerular cells. These are stimulated by the olfactory sensory neurons, and what they're going to do is inhibit all the nearby glomeruli. So only the strongly excited glomerulus communicates, and the nearby neighbors all get inhibited. So no communication here because I didn't smell that thing. Only the strongly stimulated one actually gets to stimulate its mitral cell. And then there are, of course, granule cells, right? And these are going to, again, participate in lateral inhibition. So I'm an active mitral cell. What do? I turn on this granule cell. Oh, sorry, I should have turned on the periglomerular cell too. And what am I going to do? I'm going to inhibit the neighboring mitral cells. So again, I want specificity. This is the pathway. That's a sloppy check. This is the pathway that gets turned on because I smelled whatever the heck that olfactory sensory neuron smells. Just good old lateral inhibition. Okay, so we've turned on some specific mitral cells. Those are my secondary afferents. Where do they go? Well, they're not going to the thalamus. That's the crazy thing. The tertiary afferent is not in the thalamus. Here it's in the cortex. It's in the temporal lobes. It's in that anterior temporal lobe, what we call the piriform cortex. There are some other things nearby. Yeah, absolutely. Amygdala. Entorhinal cortex, emotion, memory. These things are coming up. We're going to hear about those. But pretty much right away, before the cortex even knows what we smelled yet, we're getting some emotional response. We like it, we don't like it. We might have a memory attached to it. Maybe we like it, maybe we don't. These tertiary afferents then project, finally, to the thalamus. And then they hit the orbitofrontal cortex. And yeah, there are some connections that go around the thalamus. But yeah, we'll go through. We'll hit that, dorsomedial nucleus, mediodorsal, whatever you want to call it. So eventually we'll make it up to the orbitofrontal cortex, and this is where we have our conscious perception of smell. Not over here, not in the temporal lobes. No, here we're kind of figuring out how we feel and any memories of it, but we place the smell over here in the orbitofrontal cortex. Now, what else are we doing? Well, we're projecting into the hippocampus via the entorhinal cortex. Here's where we're probably accessing our memories. We can create some autonomic responses, right, via the hypothalamus. This would be how sense would trigger, a sympathetic response. I'm scared because some other, alpha male took a leak nearby, something like that. And we can actually project back into those olfactory bulbs to modify sensitivity. The whole idea, and by the way, these arrows are bidirectional. I know the cartoon doesn't show it. If I'm hungry, if my hypothalamus is saying I'm a little low on amino acids and carbohydrates, this can head on back, and we jump back to the olfactory bulbs, and we increase the sensitivity of those olfactory sensory neurons that pick up things that smell sweet and meaty. And when you're hungry, food smells a heck of a lot better. And why is that? It's this back propagating projection into the olfactory bulbs, making us more sensitive to food smells. And here's a nice little cartoon schematic summarizing all of that. I believe in you. I trust you. I know you can read it. I'm going to move on. But this shows everything that we've just talked about. And having done that, you should be able to handle these questions, right? We know that the glomeruli are where we're going to pick up our smells, right? We understand the lateral inhibition. We know there's feedback onto those granule cells to tune our sensitivity to certain smells. And we understand that there's a very direct projection from the olfactory secondary afferents onto emotional circuits, like in the amygdala. Let's get into memory now. So when you think memory, you should kind of think of two aspects. We're talking about episodic memory here. There's the encoding of memory, that is storing information, and that's going to happen in the hippocampus. And then there's long-term storage. That's going to be in the cortex. It's very easy to put information into the hippocampus, but it's also very easy to lose that information. It's hard to put information into the cortex, but it's also hard to lose that information. Okay, and the main place that we're gonna talk to here would be the entorhinal cortex. Okay, the entorhinal cortex is going to be our main input to and output from the hippocampus. So this is just a big old loop. What we're going to do is get input from association cortices. So here we've got some fibers connecting association cortices. That is the parts of our brain that are perceiving something. You know, you're looking at a sandcastle or the ocean or a plate of food, whatever it is, you have that perception, and that gets filtered into the entorhinal cortex. So there's my input. I then pass that into the hippocampus, and we're going to form a few synapses. Typically, we're going to start in the dentate gyrus. We'll head on over, CA3, CA1, and then we'll head on out to the subiculum here, and then finally, boop, back to the entorhinal cortex where we started, right? So it's a big old loop. And we're going to get some neurons firing together. They're going to wire together, and that's how we store that information. We remember, hey, I went to the beach and I looked at a sandcastle, because there's a pattern in your hippocampus that holds on to that. Now, if you don't recall that memory many, many, many times, it'll eventually get lost because we'll attend to something new, and well, it's easy to rewrite the connections in the hippocampus. So easy to store, easy to lose. That's what we should think of in the hippocampus. Okay, and the entorhinal cortex is our input, right? We get input from the association areas and pass it into the hippocampus. It's also our output. When we try to think, what did I see today? Oh, sandcastle. Here you go. That's what it was. So we send that back out. What did I see at the beach? I saw a sandcastle. I formed a memory, and when I think, what did I see? I can recall the memory and send it back. So two aspects. There's encoding, that's making that pattern in the hippocampus, and then there's consolidation. That's taking that pattern in the hippocampus and putting it into the cortex. Okay, so when you have that day at the beach, there's a lot of things going on. There's the feel of the sand and some somatosensory cortex. There's the sound of waves over here in this like auditory region. You can recall the shape of the sandcastle, and that's just a whole bunch of neurons being active, right? Some are active. That's the white dots here, right? We've got, looks like 9 neurons active here. Ooh, another 9, and another, I'm going to guess 9, yeah, because I'm a simple guy. Okay, so we got certain patterns of activity in these different visual, auditory, and somatosensory areas. So certain neurons in the cortex are active whenever we experience a thing. And there's a lot of different possibilities. If we just have 32 neurons, we can record, you can see here, over 4 billion different patterns with just 32 neurons. And we got way more than 32 neurons. Okay, so what goes on? Well, that pattern is going to get sent into the hippocampus, as we just went through. Now we're going to simplify it to 6 neurons. Here's what happened to me today, and I go and tell the hippocampus via the entorhinal cortex, and that makes a pattern in my hippocampus. Pretty quickly, this gets firing together and wiring together. Easy to store, easy to lose. As we study, this is what's happening over time. As we continue to recall information, these weak connections between my cortical neurons, weak or non-existent, get strengthened. Why? Because when I remember it in my hippocampus, I remember it by reactivating those original 6 cortical neurons, and they fire together. And when they fire together, you know what they do. They wire together. We'll see how in just a little bit. And if you study something enough, or if you say something enough times, eventually your cortical neurons get wired together. And if I were to ask you your name, I bet you could tell it to me. And I bet you'll almost never forget your name. Why? Because this is now very stable. Okay, so that's the trade-off. Hippocampus is very much plastic. That's not stable. It's easy to change, but the cortex, very stable. It's hard to change. That's good and bad. It's hard to form stable memories, but once you do, it's hard to lose them. That's why we have to study many, many times before we actually become an expert in something. Okay, so let's now go into the hippocampus proper. That'd be this little seahorse-shaped structure right here. The hippocampus, as you can see, kind of shaped like a seahorse. Some people think it looks like a jelly roll, whatever works for you. This is an older form of cortex with only 3 layers, so it's not neocortex, it's allocortex. And we find it in the medial temporal lobes. Okay, this is why when we have damage in the medial temporal lobes, we have severe memory impairments, because that's where the hippocampus lives. 3 general areas to think about. First and foremost, there is the dentate gyrus. That's this pink area here. The dentate gyrus, which has the granule cells, some newborn neurons. Then we've got this blue structure over here. That's the cornu ammonis, or Ammon's horn. CA, much easier to say. There's CA1 to 4. 4 is closest to the dentate gyrus. 1 is closest to the output. Look, CA1's over here. They highlight CA3 because this is where we have most of our synapses happening from the dentate gyrus. And then finally, we got our output, another pinkish area, the subiculum. And that's the order of events, right? We're going to start off in our entorhinal cortex. There's the start. We're going to project into the dentate gyrus. That's step 2 there. From there, we project on over into CA, usually CA3. From there, we're going to form another synapse, CA3 to CA1, called the Schaefer collateral. And from there, we output to the subiculum. We've seen this before. Where do we go from the subiculum? Well, mostly back to the entorhinal cortex. We're making a loop. And we do that so that we get certain neurons firing together in the hippocampus, so they wire together, so we store some information. Okay, so in connections with the hippocampus, largely from the entorhinal cortex. And we can target the dentate gyrus, or we can bypass that and just go to Ammon's horn over here. 2 different pathways. The perforant path, as you can see, perforates through Ammon's horn to get to dentate gyrus. The alveolar path follows along in this fiber tract called the alveus. So we're not perforating through. No, we're going to go around, and we're just going to synapse in Ammon's horn. We're going to bypass the dentate gyrus. This is just a way of getting 2 very different patterns without having to have a whole lot of extra brain structures. So we can have a pattern that includes the dentate gyrus via the perforant path, or we can exclude it via the alveolar path. And then, of course, we get to the subiculum and we head back to the entorhinal cortex. Okay, so we got some input from the entorhinal cortex and we provide output to it. I know I've said this a few times, and there's a reason. When we repeat things, we tend to remember them. Okay, so that's one kind of connection we can have. We can also link up with all kinds of other limbic structures via the, I think it's called Papez circuit. Maybe it is Papez. No one actually can really figure that one out. That's the hard part, is just pronouncing it. Okay, so we start off here in the hippocampus, and what are we going to do? Well, we're going to follow this fiber bundle called the fornix. Let's highlight that. So the fimbria is the fornix on just one side of the brain. This will come together and form a midline structure. So instead of sending output to the subiculum and getting input from the entorhinal cortex, no, we can get input via this fornix and provide output too. Okay, so the hippocampus here, we're going to project along the fornix, hitting the mammillary bodies. Then we have the mammillothalamic tract. That's going to hit the anterior thalamus. Calling back to the last lecture, from the anterior thalamus, we head on up to the cingulate cortex. Hey, that's the cortex, by the way. And this is a part of the limbic system. So we can figure out how we feel about a certain thing, and then the cingulate can send that back to the entorhinal cortex and back to the hippocampus. And look at that, it's a big old loop. So it could be a loop that's really just entorhinal cortex, hippocampus, and it could be a loop that involves more emotional structures. Okay, so what does the hippocampus have to do with memory? Well, I'd say everything. So what we're seeing here is synaptic plasticity that occurs with learning. So they put in a few electrodes here, looks like about 8 if I can count that correctly, and they have a stimulating electrode. So they stimulate those fibers from CA3 to CA1, and they're recording what's the response. In other words, how strong is the synapse? Here's the raw data that you really don't care about. Let's just go with this color-coded heat map over here. The redder it is, the more of an increase. If it's blue, it didn't really change, and if it's purple, it got weaker. Okay, we see a whole lot of reds, yellows, increases in strength here in the trained rats. And what they did was put them into a cage and shock them. And they quickly learned this cage sucks. And that's all they had to learn. It's very easy for rats to learn this. Notice, massive increase is in synapse strength. So when they put the rat in the cage, they see, do they remember that this sucked? Yeah, absolutely. Right? They have a memory of it here. Those that have learned to avoid that cage, those that were trained, have a change in hippocampal synapse strength. If you just let them walk through the cage and don't shock them, or if you just shock them and you haven't put them in a new cage, they have nothing to learn. It's just, what the hell, why did you shock me? Or you don't do anything to them at all. We don't see major changes in hippocampal strength over time. No, we see a massive change whenever they had something to learn. You put me in a new cage and you shocked me. Boy, that sucked. I learned I don't like this new cage. So the degree of learning is related to the degree of synaptic plasticity, which for our purposes just means that increase in synapse strength. Okay, when we talk about synaptic plasticity, what we mean is long-term potentiation. This is the same thing as that good old Hebbian plasticity we've talked about time and time again, which, to summarize it, is cells that fire together, wire together. In other words, fire together. Co-activation of pre- and postsynaptic neurons. They're firing together. Strengthens their synapses. They wire together. And here it is right here. There's learning. Look at that. The sexiest plot you've ever seen. So here we are. We're looking at synapse strength. 1 just means normal. So here's what it is beforehand. And then they give a really strong stimulus to the hippocampus. And boom, we see, wow, now the synapse is like 40 or 50% stronger. So not quite doubled, but a heck of a lot stronger than it was before. Why? Because cells fire together and then they wire together. How does that work? The NMDA receptor. And we know the NMDA receptor is important because if you put on AP5, you get a little short-term increase, but it just doesn't last. You don't form a memory when you antagonize NMDA receptors. So the beauty of NMDA receptors is that they have the magnesium block. If I'm getting input from this presynaptic neuron, but I'm not firing, the only thing that gets stimulated is the AMPA receptor here. My green NMDA receptor is still blocked by magnesium. It's not until I'm firing as well that I depolarize enough to get rid of the magnesium block. So now, when I fire, I get rid of magnesium, and when the presynaptic neuron fires, it spits out glutamate. What happens? Sure, I'm still turning on AMPA receptors, but importantly, I'm turning on NMDA receptors, and they bring in a ton of calcium, which turns on some kinases. I don't think I care if you know them, but in case you care, protein kinase A is going to stimulate the insertion of additional AMPA receptors. Look at that. I go from 1 to 2. And CAM kinase 2 phosphorylates AMPA receptors to make them move more cations into the cell. So even though I might have the same amount, they move more ions. In other words, I have increased conductance and abundance of AMPA receptors. I have a stronger synapse. And that's why I see this. I see a stronger response in that synapse after LTP. Now we can modify how well LTP works with some other inputs. For example, we could provide additional glutamatergic input via the amygdala in case we're afraid of something, right? If something is emotionally salient, we're more likely to remember it because the amygdala provides more excitatory input, so pre- and postsynaptic neurons are more likely to fire together. Same thing for the septum, so if we're awake, we're also more likely to remember things. Okay, cool. So it could be some dumb old glutamatergic input, just exciting pre- and postsynaptic neurons. It might be something a little more exotic. Neuromodulators, for example, acetylcholine, norepinephrine, serotonin, dopamine. These are going to act on G protein coupled receptors, for the most part GS, but there's some GQ. So we can activate protein kinase A, right? That's what GS is going to do. It'll also get CAM kinase 2, I promise. And GQ is going to be a whole lot more. Wow, that was the least informative thing. I meant to write protein kinase A. Okay, there we go. And GQ is going to stimulate CAM kinase 2 by bringing in all that calcium out of the endoplasmic reticulum. We turn on those kinases that we did in the last slide, and what do? The same thing they did before. Let's insert more AMPA receptors. Let's make sure that when they're there, they move more ions. And guess what? We can go and modify gene expression. We can turn on Krebs, and what do? Hey, let's make more AMPA receptors, and let's build bigger synapses. And so if we have, something like, emotions, is something rewarding? If so, dopamine comes on, and we're more likely to remember it. Right? If something is, emotionally arousing, we might get some noradrenergic input as well. Now, the effect of stress is a little complicated. It depends. If we're talking acute stress, we're more likely to remember that thing. Hey, something tried to attack me, I should probably remember that. And so there are two types of receptors that glucocorticoids are going to act on. There's the mineralocorticoid receptor, and these things seem to promote LTP. So if you let a rat do something stimulating like swim, if you let them swim, what we see over here, we let the rat swim, we do the whole LTP thing, and we find, the swimming enhanced LTP. Why? Because it was moderately stressful, right? And it was stimulating, it happened the one time, and when we have acute stress, or what we might call stimulation, that promotes LTP. And it does it by activating mineralocorticoid receptors. How do we know that? If we block them over here, we don't get the LTP anymore. Yeah, we get the short term, but it's lost. Okay, so mineralocorticoids actually promote learning from stress. So we remember those acute stressors, and we can better respond to them, hopefully. What about long-term stress? Well, now we switch from activating mineralocorticoid receptors to activating glucocorticoid receptors. And this is what happens in chronic stress. This would be things like, the rat's getting handled. Okay, that's stressful and it's not as stimulating. And what we see is that handling the rat, they don't really like that. That actually impairs LTP. Look, we're cruising along here at 100%. We stimulate LTP and we don't get it. We're not getting LTP whenever the rats are handled. And why? Because they're stressed. And we handle them many times, and so this chronic stress prevents LTP. We can prevent it by antagonizing glucocorticoid receptors. Take that same handled rat, right? Here we are. We try to give it LTP. It doesn't work. Take the same handled rat, antagonize glucocorticoid receptors, and now we can get LTP again, right? Here we are, normal synapse strength. We stimulate the heck out of the hippocampus, and now the synapses are way stronger. We get LTP. So stress can make us remember a thing acutely, but chronic stress prevents LTP and therefore interferes with memory. And so this linking up of emotional and stress circuits with memory circuits makes absolute sense. We want to remember what made us feel good. We want to remember what stressed us so we can go toward the good things and avoid the bad things. Whenever we have damage to primarily the hippocampus, the medial temporal lobes, we have major problems with memory. And I'd say the most famous patient, the most famous neurological patient is patient HM. And here's a little portrait of him. He had severe epilepsy because of a childhood traumatic brain injury. And to treat that, they removed large portions of his medial temporal lobes. And we know what lives there, the hippocampus. So they removed both of his hippocampal complexes, and he couldn't remember a thing from then on out. He remembered all the things that happened early on in his life, a couple years before the surgery, but he never formed any new memories. And he worked with the same neurologist for the rest of his life. Every time he met her, it was like he was meeting her for the first time. Okay, this is an old classic case. More recent case is a fellow named Clive Waring, and he developed not epilepsy, but he had herpes encephalitis. And herpes virus, when it gets into the nervous system, affects very similar regions. In addition to the medial temporal lobes, we're also hitting some frontal lobes, right? So we're going to get a little extra damage, but key point, we are going to severely damage the hippocampus. And what happens here?
Clive really only has less than 30 seconds memory. And sometimes it's as little as perhaps 7 seconds. It's as little as a sentence.
I'm going to see your sister, Adele. Her daughter's got married recently in New Zealand. And so they're having a party.
Funny how the ladies acquire different title when they get married.
Do you know who I'm going to see tomorrow?
Buckingham Palace?
No, really guess.
I don't know.
You don't know? Adele.
Oh, I see.
Do you know why I'm going?
No.
She's having a party at her house tomorrow.
It's her birthday, isn't it?
No.
No.
Do you know why?
No.
It's to do with her daughter.
I see.
Do you know why her daughter's having a party?
No.
Guess.
No, I don't.
She's just got married.
Oh, I see.
She just got married in... Do you know what country she just got married in?
No, I don't.
In New Zealand.
Oh, I see.
Yeah.
The sentence he is in, he will probably have forgotten the sentence before. You ask him a question and he'll give you an answer, but while he's giving you the answer, he's already forgotten the question.
So as you can see here, without a working hippocampus, Clive can't keep track of his day, and he can't keep track of any new information. So it comes in, he can hear the words, he can understand them, but when it goes into the entorhinal cortex to try to make some new pattern in this non-existent hippocampus, it fails. And so you'll notice his wife there tells him that his sister's daughter is getting married, and he's forgotten it at the end of the sentence because he could never store it in the first place. Okay, so make sure you're reviewing that stuff. You want to make your neurons fire together enough times that they're going to wire together, and you can answer some stuff on the exam coming up. So make sure we're getting some idea of, we have some perception and we want to remember it, right? We go from association cortex to entorhinal cortex into the hippocampus. Those neurons fire together and wire together via LTP. Make sure you understand how that thing works. And then we have a pattern in our hippocampus. Now remember, hippocampus is connected to entorhinal cortex, which goes back to those association cortices where we had the perception in the first place. And so when someone tells us that our niece is getting married, the cortex perceives it, but the hippocampus stores it. And so when someone asks us, how's our niece doing? We can recall she's getting married. One last thing to hit here, and it's a doozy. This would be emotion. Of course it's a doozy. We've got a lot of emotions, and emotions create these really profound changes in our mood, and they change our behavior, and that's the whole point of them, to modify our behavior. Again, that's why olfactory circuits are going in here. The first animals that were slithering around in the world, if they smelled food in one direction, they should turn toward that direction. And so maybe they don't have an emotional response, but they do have a behavioral response. Now, who the heck knows? Maybe nematodes do feel a certain way about salt and peanut butter. But, the complex emotions that we have still serve the same function. change our behavior. So hopefully our emotional circuits are wired into behavioral circuits too. Otherwise, maybe I have no idea what I'm talking about. Okay, we're going to hit through a laundry list of areas, and then we're going to wire them together in the end. All right, first one we're going to think about is the anterior cingulate cortex. We're in the cortex, and when we think cortex, let's think positive, okay? The anterior cingulate, it's got a lot of areas. No doubt about it. Is it just for positive emotion? No. It's also there to control your bladder and also there to help you speak. So we can get a lot of problems if we have damage to the anterior cingulate. In addition to emotional problems, right, we can have problems moving, we can have problems speaking, we can become incontinent. So it does a lot of things. I'm not saying that the anterior cingulate cortex is only there to make you feel good, but that is one of the things that it does. So please, bear with me when I vastly oversimplify the anterior cingulate and say that it's indeed active during positive emotion. And for the most part, it's not doing much when we have negative emotions. That is in most folks. So here we're looking at healthy controls, that's the HC, and we're looking at activity right here in the anterior cingulate cortex. And here they're showing them neutral stimuli. We shouldn't see much going on. In fact, we don't. We're showing negative stimuli, so pictures of people getting hurt. And we're not seeing an increase in activity. Because again, I want to oversimplify and say that the anterior cingulate is all about positive emotion. That's when it's active, okay? It is an oversimplification, but please allow it. Then we have two other groups over here. Folks with schizophrenia who are not prone to violence and folks with schizophrenia who are prone to violence. And notice the one key difference. You show them pictures of people getting hurt, and what happens? They have more activity in their anterior cingulate cortex. And so to them, violence isn't a negative thing. Violence is happiness. And so why are they prone to violence? They just simply can't help it. When they think of violence, they don't think of it as bad. It's positive to them because the anterior cingulate is active. And that is, in our simplified world, what makes us feel positive. What makes us feel negative? The amygdala. Is that an oversimplification? Yes, of course. But we're going to take it. So when we think amygdala, we're going to think negative emotion. Okay. So the amygdala is linked up with a number of other limbic regions, the hippocampus, for example, as we already talked about. Hey, if something's really emotionally salient, we should remember it, especially if it's something that was going to harm us. That's worth remembering. I got attacked over there. Let's avoid that part of the jungle. It's linked up with the insula. The insula is the region of our brain that makes us feel disgust. So this is why, it's also activated by pain. This is why whenever we don't like a thing, something makes us angry, we also feel just simply disgusted by it. We're not reinventing the wheel here. We're just going to repurpose physical disgust with emotional disgust. That's what's going on here, right? Also, there's a link between the insula going into the amygdala. So when we're in pain, how do we feel about it? Not very good, because the insula activates the amygdala, and we get negative emotion. Okay, how about the nucleus accumbens? Well, we have, of course, inhibitory links here, because the nucleus accumbens is the thing that's going to turn on the anterior cingulate. So when my nucleus accumbens is active, I'm going to go turn off the amygdala. So when I feel good, I don't feel bad. That's that whole winner-take-all reciprocal inhibition between positive and negative emotion. When you're happy, you're not sad, and vice versa. So here we can see some data showing us the activation of the amygdala as well as the hippocampus. When we're learning, recalling some autobiographical information, we see certain parts get activated when we're recalling a memory, and just some semantic information over here, the gray one. Now, mostly we're going to get activated with autobiographical information because there's a lot more emotion involved with that. It's easy to remember stuff about ourselves. We are the most important thing to ourselves. And they're just separating the anterior and posterior portions. No big deal there. Who cares? We're seeing a spike in either case. Doesn't matter. But notice, hippocampus, down here, and the amygdala are both activated during memory, because we probably have some kind of an emotional link with our autobiographical memories. Are they all negative? No, and that's why it is an oversimplification to say that the amygdala only deals with negative emotion. Largely it deals with it, but it does other stuff too. I would say it's more emotional salience. Is there any emotion at all? Yes. When I think back on my life, I have emotions. I'm proud of some things, I'm disappointed in some things, and it's probably that amygdala activation that's giving me that. Now, of course, the amygdala gets input from association cortices as well, right? Oops, sorry about that. The association cortices are going to create some perception, and then we're going to have a feeling about it, right? We see somebody approaching us, and they have a smile on their face. Hey, we're not afraid. They have a scowl on their face, and they look very angry to see us. We probably are going to need to feel a certain way about that. So when we see negative things like people getting hurt or angry people, those perceptions created in association cortices funnel directly into the amygdala so that we can create an emotional response to our perceptions. And then the amygdala is also linked up with a bunch of autonomic areas. The locus ceruleus, right, to wake us up. Hey, someone's angry and about to attack us. Maybe I should wake up and do something about it. Oh, I should also probably increase my heart rate so again, I can do something about it. And boy, wouldn't it be nice if I triggered a stress response so again, I can do something about it. So the emotions are there to, again, change our behavior. So if we have some kind of negative emotion, what do we need to do? Well, we need to wake up. Great, handled. We need to move more blood. Handled. And we need to make sure we're liberating some glucose from the liver and we're moving that, blood around, getting that stress response. Handled. So the amygdala is well connected. It's talking to basically all of the cortex, it's talking to the rest of the limbic system, and it's talking to our autonomic nervous system. It's hard to find an area that isn't hooked up with the amygdala after a couple of synapses. Now, the whole purpose of the amygdala is to identify emotion, and in particular, negative emotion. So we see most activity in the amygdala when we look at some kind of a fearful or angry face. So here we have fearful voice, fearful face. That's where we get the biggest increase in amygdala activation. And that's just these little dots over here. Again, medial temporal lobes a little in front of the hippocampus. They're next door neighbors. That way emotion and memory are right there next to each other. As we reduce the amount of fear present, so we hear a fearful voice, we see a happy face, okay, the fear gets me going a little bit. We see a happy face, fearful voice, okay, same thing. Everything's happy, a little blip. That's just to probably say, hey, it's not negative. No, we get the biggest response when there's negative stuff, because I need to probably do something about this. If everyone's happy, I probably don't need to turn my heart rate up, move the blood around, things like that. So we don't have patient HM, but we do have patient SM. And patient SM had a congenital disorder that caused bilateral degeneration of her amygdala. So she had no amygdala. And here it is. Oh, I guess I should say here it isn't. So we should see an amygdala there. There it is. Here it is, absent in SM. Sorry, this is the hippocampus. Here's the absent amygdala, so they move forward a little bit in the brain. And patient SM couldn't recognize dangerous situations. They found this out because she kept winding up in the emergency room because she would get attacked. She wouldn't avoid bad areas, dangerous-looking groups of people. She'd get attacked, mugged, end up in the hospital. So they did some imaging, and they found out she doesn't have an amygdala in there. And that's what's going on there. She couldn't recognize negative emotion, and she couldn't adjust her behavior accordingly. So negative emotion is still important, even though it sucks. And heck, animal studies show the same thing. You'll see two different rats. They're going to come out pretty soon. There they are. Okay, and they're going for these food pellets right there. And there's a robotic cat, and you can see one of the rats is appropriately scared. The other one couldn't care less. Walks up, gets its food, heads on back. Now, the video's running. You can check this out in your own time. It's going to run for another minute or so, and then the rat's going to try to peek out. It's going to come out, and the cat will scare it again. Sorry, spoiler alert. This rat is definitely afraid of the cat because it has an amygdala. This rat down here, no amygdala, no fear. And it worked out in this case, but in nature, that cat's going to eat it. Now, the amygdala, if it runs amok, creates a problem. And what we find is that people who have anxiety are people who have more pronounced amygdala responses. So folks who have normal levels of anxiety, right here, they're looking at angry and fearful and happy stuff. And there's some changes in amygdala activity, sure, but nowhere near the same amount as we see in anxiety-prone subjects. And here, oh, by golly, we're seeing this massive increase in blood flow in the amygdala. And so their amygdala is more active, and therefore they're more likely to have negative emotion. And that's all anxiety is, this negative emotion. In this case, brought on by a hyperactive amygdala. Okay, so we got the anterior cingulate. That's going to be positive. We feel good. Amygdala, we feel bad. The nucleus accumbens is where we're kind of deciding, do we feel good? Do we feel bad? Is this thing going to be rewarding? This is where kind of decisions are made, and that's going to be based off of whether or not we're getting dopamine input or whether or not we're getting glutamatergic input from the amygdala. Okay, so the nucleus accumbens is also called the ventral striatum. Not the dorsal striatum, that's lecture 19 stuff. But just below it, the ventral striatum. And so it also has spiny projection neurons. And hey, guess what? We've got D1, we've got D2. So there's going to be different effects of dopamine on these different spiny projection neurons, and they have different targets. Yes, I'm oversimplifying, but we have basically the direct pathway. Oh, nice. This is going to drive activity. And then we have what we'll call the indirect pathway, and this will drive inactivity. It's going to make us feel defeat, depression. We're going to be less likely to do stuff. So if we have a whole bunch of negative emotion and there's nothing we can do about it, the best thing to do is to just do nothing. And that's what depression is. This feeling that I can't do anything, there's no way I can get out of this bad situation, and that's what we're going to make here. Very negative mood. So it depends on which pathway is active. And what you'll notice here, dopamine's going to turn on one of them and turn off the other. In other words, we're going to do things and we're going to not feel depressed with dopamine. What's going to go on with the amygdala, on the other hand? I will make the dopamine green. The amygdala releases glutamate, and that turns them both on. So what are we going to do? We're going to do something, but we are not going to feel good about it. Okay, let's have a look at what's going on in these different spiny projection neurons. So what we have here, we've got a little rodent, and we put a little light emitter in its head. So we put a fiber optic cable in, and we can shine blue light. And what that blue light does is turn on neurons that express this light-sensitive cation channel. It's the same one we mentioned in the whole thalamus lecture. This one turns on neurons. Okay, so what are they doing here? Here they're going to stimulate different neurons. There's the D1 getting turned on. That's what's going on here. And then there's the D2 getting turned on. Okay, what this rat is learning is something very simple, and that's that if it goes over to this part of the cage, it gets cocaine. And rats, just like humans, really like cocaine once they experience it. And so what we're going to do is give them not quite enough cocaine over here in panel B. Not enough to get anything going, and so they're not really feeling it. It's a less than effective dose of cocaine. There's some, but not enough, right? They need more. But if we supplement the cocaine by stimulating D1 neurons, then they learn to spend more time over there in the chamber. Okay, so the y-axis here is showing us how much more time they spend in the party room, the one where they get cocaine. Now, when you don't get a whole lot of cocaine, you don't learn that that room is any better than the other, and you can see we're pretty much not spending any more or less time in the cocaine room. However, we're going to spend a whole lot more time in the cocaine room if we're stimulating D1 SPNs. And why is that? Well, when I'm over here, for some reason, it just feels right. When I think of going over there into the cocaine room, my DSPNs inhibit the output of the basal ganglia, so I don't not go there. In other words, I'm driving activity to go to the cocaine room. Okay, what is D2 doing? It's not driving activity. So here I am, I'm giving more cocaine. Notice, we're giving 50% more cocaine. And if I don't do anything fancy, notice rats really like to hang out in the cocaine room. We're nowhere near zero. These rats, just normal rats, are hanging out over here because that's where they get their cocaine. If, while they're getting their cocaine, we stimulate D2, that is, the cocaine is inhibiting them, but we're using light to activate them. So if we turn on the D2s, the rat's over here, it's getting cocaine, but it's also getting these feelings of defeat and depression. And so you'll see, yeah, it's spending a little more time over in the cocaine room, but nowhere near as much as it would have if D2 wasn't active. So what these are showing us, stimulate D1, you're more likely to do that thing. Stimulate D2, you are less likely to do that thing. Okay, here we're using a fiber optic cable. That's not what happens in real life, right? Oh, here's the rat, by the way. There it is going into the party room. Boom. Those D1 neurons get turned on. D2 turned off. All right, this will play again. And notice when we're walking up here, D1's going to get turned on and D2 turned off. Why is that? Because we walked into the cocaine chamber and we expect to get some cocaine. And so we get dopamine input, turning on D1s and turning off D2s. So we do a thing and we don't feel bad about it. Now, what makes us feel bad normally would be, well, something like this. This is what we call the social defeat model. So they put this smaller mouse in with the bigger mouse. So we got the little black mouse, the big white mouse, and the big white mouse just kicks the little black mouse's ass all day because mice suck. Maybe you feel bad about animal research until you work with them and you realize if they could, they would eat each other's face. Even their brothers and sisters, they'll do that. So mice are little assholes, and when you put a bigger mouse with a smaller mouse, the big one beats up the small one. Because again, like I said, they're assholes. And so what they do, they let this little mouse get beat up, and it's really horrible to do that. And even worse, he's going to go live right here. So this little black mouse is going to have to come over here, and here's where he's going to spend his days, and he's going to be over there. He's going to be smelling his rival. He's going to be seeing his rival. He's not going to feel very good about this. This is the social defeat model that we're getting these data from. So what goes on in that poor little black mouse's head? Well, we find that some mice really don't care. You kick their ass and they pick themselves off and they dust themselves off and it's fine. But then there are those who are susceptible to this social defeat. That is, they become depressed. They don't do anything anymore. No thanks. And when you look at the amount of activity that they get on their D1, this should be spiny projection neuron. We know what they're called. The old word is medium spiny neuron, but we know they're spiny projection neurons. We see less input. In other words, there's a decrease in D1 spiny projection neuron activity. That's why they're less likely to do stuff. That's why they seem depressed. They don't want to move around. They've given up. What happens? D2, more input. They're more active. And so what's going on there? There's more D2 output, and so they feel like shit. They don't do things, and they don't feel good. And that's because of the social defeat. That lack of dopamine is what's creating that. So as we can see here, dopamine is going to be a pretty powerful regulator of our mood, and as we covered in Lecture 19, whether or not we do a thing. And that's, of course, via the ventral striatum and dorsal striatum, respectively. So, all of those addictive drugs act on dopamine either directly or indirectly. And for the most part, the more directly it acts on dopamine signaling, the more addictive it is. Cocaine, amphetamine. These directly act on dopamine signaling by inhibiting or reversing dopamine transporters, and therefore pretty well directly increasing dopamine levels. So when we use cocaine or amphetamine, we get an increase in dopamine, and what happens? We're more likely to do those behaviors, and we feel good about them. When we don't do them, we feel bad. Nicotine acts on nicotinic acetylcholine receptors on dopamine neurons. That's this brown cell here. Heading into the dorsal striatum, the ventral striatum, wherever, doesn't matter. When we use nicotine, what do? We directly depolarize dopamine neurons, so they spit out more dopamine. And in case you haven't heard, cigarettes are highly addictive. Okay, we can also indirectly increase dopamine input. These are still highly addictive, but they're going to act on different neurons. Okay, opioids, what are they doing? Well, they are inhibiting inhibitory neurons. Good old disinhibition. So I take an opioid, this stimulates those mu-opioid receptors, so this inhibitory neuron is less active, so I'm not inhibiting the dopamine neuron, so it's more active. Okay, indirect, but the same thing as nicotine. Sure, nicotine is going to depolarize me. Opioids are going to disinhibit me. What's the net result? I spit out more dopamine. Ethanol, same thing as opioids, shorter time period, but still highly addictive. What happens? I'm acting on GABA receptors in this case. Same thing though, they're still inhibitory. So I'm inhibiting an inhibitory neuron. In other words, I am disinhibiting this dopamine neuron. And then cannabinoids. Cannabinoids are going to act presynaptically. And what they can do is kind of a couple of things. They can act on inhibitory neurons and make dopamine neurons more likely to spit out dopamine. They can also act on the dopamine neurons themselves, right? Decreasing dopamine release. So it's kind of a complicated story. And we tend to see that cannabis is not as addictive as other drugs, although, hey, everything can be addictive. So, be careful out there. So what's the problem? Well, what we're doing here, as you can see, we're increasing dopamine, either directly or indirectly. Okay, don't forget homeostasis, right? Long-term use, what are we going to do if we keep stimulating dopamine receptors? We're just going to downregulate them. Okay, fine, you keep stimulating the heck out of your dopamine receptors. I'm just going to get rid of dopamine receptors, and I'm going to have lower levels of them, and that's what we see in folks who abuse these drugs. For example, cocaine and methamphetamine abusers have lower levels of dopamine receptors in there, in this case, caudate, in the striatum, in other words. Controls, you can see the red there. That just indicates a higher density of dopamine receptors. Notice we're losing some of the red. Oh, this isn't as red. Why? I have fewer dopamine receptors. And so when we're not on the drugs, what's going on? Well, we don't have as much dopamine around, so our mood is going to lower. And when we think about using the drug, that is going to trigger dopamine release, because that's what dopamine is all about. Sure, reward triggers dopamine release, but when we expect a reward, that also triggers dopamine release. So if you're addicted to a drug, just simply thinking about the drug stimulates dopamine release. And thus we feel a little bit better. But we're also more likely to carry out the behaviors that make us go and get and use the drug. And so addiction is really happening under the hood, outside of conscious control. And once you're addicted to a substance, you just simply can't help but go seek out that substance. Okay, so our emotions are there to drive our behavior. They're also there to help cue in on memories. Right? Do we remember a person? Well, do I have an emotional response to him? If I've met a person before, we've probably interacted. Maybe it was positive, maybe it was negative. But I know that I know a person because I have an emotional response to them. And the same thing seems to be true for our homes and our trucks and things like that, because when we have damage that severs the connections between the limbic system and the cortex, where our emotions get separated from our perceptions, then we think everything is an imposter. And that's what we call Capgras syndrome. Here's an example here.
2 years ago, David was involved in a terrible car accident while driving back to California from Mexico.
There was a problem with the car, and I landed in the highway with my head first.
Okay.
Like this truck that is coming by?
For 5 weeks, David lay in a coma. Serious injuries led to the loss of his right arm. But to everyone's relief, when he regained consciousness, his mental capacities seemed to be intact.
He was articulate, he was intelligent, not obviously psychotic or emotionally disturbed. He could read a newspaper. Everything seemed fine, except he had one profound delusion. He would look at his mother and he would say, "This woman, doctor, she looks exactly like my mother. But in fact, she's not my mother. She's an imposter. She's some other woman pretending to be my mother.
All right, we can check that out in our own time. Okay, so let's try to put all of that junk together. So here's the, I promise, simplified limbic system model. So part of it is going to affect behavior, right? We're going to affect that whole basal ganglia output, affecting the motor nuclei of the thalamus, talking to the motor cortex. So we're more or less likely to do a thing. Well, it depends on how we feel. And how we feel about a thing is kind of put together in the ventral striatum. Am I getting more dopamine input? Am I getting more input from the amygdala? And based on that, I'm going to be more likely to do a thing, or maybe I'm going to feel a certain way. Okay, we've got our memory circuits over here. We've got, when we're in pain, the insulas in there, right? If something hurts us, we hopefully remember that, and that makes us not feel good about it, so we're less likely to do that thing again. Okay, lots of things in here. Spend a little time on it. Let's go through a few examples. Let's say something good happens. Okay, if it's good, we're not getting a lot of amygdala activation. Hopefully we remember that it's good, right? We perceived, hey, this is good. This is likely to be rewarding, and so we tell the entorhinal cortex about it, and we store a memory, and the hippocampus can go and talk to the ventral tegmental area, and so does the association cortex. So when we're feeling good, we're activating those dopamine neurons in the ventral tegmental area, not the SNC, that goes to the dorsal striatum. The ventral tegmental area goes to the ventral striatum, that is the nucleus accumbens. And what does that dopamine do? It turns on D1 SPNs and turns off D2s. So we don't inhibit positive emotion. In other words, we are feeling good. There's the anterior cingulate. There's another part of the cortex, the orbital frontal cortex. These are going to work together to make you feel good. And what else happens? We do inhibit the output of the basal ganglia. So we're more likely to do stuff. And you probably noticed this. When you're feeling good, you're more likely to get out there and talk to people and do things, right? If something is scary, okay, particularly scary, the amygdala's turned on, and sure, we'll get all this like autonomic respiratory stuff. Great. That's wonderful, right? We perceive something as, oh my God, it's scary. Ah! Okay, so that goes into the amygdala, and it figures it out. There's a couple of synapses in here. Who cares? The output of the amygdala triggers those automatic responses. We're going to stimulate the ventral striatum with glutamate. We're also going to then act on the hippocampus. Hey, please remember this. Also, don't feel good about it. Be a little disgusted. This sucks. Go to the entorhinal cortex. Make that whole loop. Don't activate my ventral tegmental area. No, instead activate these inhibitory neurons here. So I don't provide dopamine input. So in the ventral striatum, now notice the balance is different. Not as much dopamine, but more glutamate. So I will inhibit the output. So I'm going to do a thing. Run. But I'm not going to feel good about it because I'm also going to inhibit, via those D2 SPNs, positive emotion. Okay. Something sad. It's not necessarily scary or anything like that. I'm reducing dopamine input and I'm reducing glutamatergic input. I'm not scared. I'm sad. This is depression in this case. So in that social defeat, right, in depression, what's going on? Largely it's just D2 SPNs that are active. So I don't inhibit the basal ganglia, so they make me not want to do a thing. Right? I am not motivated to get out there. When you're sad, you don't want to do a thing. You'd rather lay in bed. You damn sure don't want to go talk to people and have them ask you how you're doing. And your D2 SPNs make sure you don't feel good about it. Now, you might think sadness is going to involve the amygdala. Actually, sadness seems like a profound decrease in activity across the cortex. And in depression, what's going on is we have decided our situation is so bad, there's nothing I can do about it. And so the best thing I could do is just not do a thing and not waste my energy. Okay, how about drugs of abuse? Well, acutely when we use those drugs, whether it's nicotine turning on this dopamine neuron, or it's cocaine or methamphetamine that make the dopamine hang around longer, or we've got cannabis or opioids or ethanol or benzodiazepines, any GABA agonist inhibiting that inhibitory neuron, we're getting more dopamine input. And whoa, this is likely very rewarding. Oh, hey, guess what? Even if you think it's harmful, those GABA agonists, those opioids, we're going to turn off the output of the amygdala. So Gotcha. Head you off right at the pass. You think it's harmful? No, I don't think so. Nope, we're not going to perceive that it's harmful. Yes, it's ruining my life, but hey, the amygdala disagrees. So what happens? We're not stimulating over here with glutamate, but we are stimulating the heck out of the ventral striatum with dopamine. And what's the deal? We feel great. And we are very likely to do stuff because we inhibit the basal ganglia. So they don't inhibit movement. What about in the long run, though? Okay, what's going to happen? Anything that we inhibited becomes overstimulated, and anything that we activated gets downregulated. And so now, hey, you kept all that dopamine around. I'm just going to downregulate my dopamine receptors. So I no longer excite the D1 SPNs. I no longer inhibit the D2 SPNs. Okay, what am I going to do? Well, I'm also going to have more amygdala output. I don't feel very good when I'm not on that drug. And I know that's what's causing it. I've got a memory. Hey, if I could only get that drug, boy, wouldn't that be good? That might trigger some dopamine release. But I'm getting a lot of excitation here in the ventral striatum. And so what happens? Now I'm highly motivated to do a thing that is go get more of the drug. And I do not feel good until I go get that drug. And what do you control in this whole equation? Maybe this. Okay, and that's a huge maybe. Maybe you control the cortex. Do you control your ventral tegmental area? No. Do you control your ventral striatum? No. Amygdala, hippocampus? No. You don't control pretty much anything in here. Maybe you have a little bit of a vote in the cortex, but where all the decisions are being made, you have absolutely no conscious say. So it's best to not overindulge, right? Everything in moderation, because you cannot escape homeostasis. And once you have an addiction, the thinking is that you pretty much have it for life, because you never really undo those homeostatic changes and those memories that you form, those associations between the substance or the action. If you have a gambling addiction, right? Whatever you're addicted to, you are addicted to forever. And you just simply have to avoid it. You can't avoid this stuff, though, so make sure you review this. We want our amygdala to be negative emotion. We want anterior cingulate to be positive emotion. And we want the nucleus accumbens to be kind of that decision maker. How am I feeling? Am I feeling good? Am I doing things? Make sure you get an idea of what's going on there, how they're connected. I hope that crazy diagram in the last couple of slides helps you with that. Make sure you're getting an idea of how the amygdala is linked to both emotion and memory. Right? Who am I talking to? Directly or indirectly, I'm going to inhibit the positive stuff, right? I'm going to activate those D2 spiny projection neurons to inhibit the anterior cingulate. I'm going to stimulate the hippocampus so I remember bad things. And then, of course, pain, insula. Why doesn't it feel good? Because the insula stimulates the amygdala, and then the amygdala inhibits positive emotion. So when we're in pain, hey, we don't feel good. Now, if there's anything that you don't feel good about and you'd like me to review in class, you know what to do. Use that questions box, and I'll make sure we get it handled. I'll see you later.